tried to get my Mac to work, but it didn't, so it's... Are the slides, are they there? Just a second. It's nice to see everybody. My name is Zach Grinspan. I'm a pediatric epilepsy doc at Weill Cornell Medicine. I direct pediatric epilepsy research, and I'm vice chair of health data science there. And I'm going to talk about this trial you've heard about of Ravicti, this drug, glycerol phenylbutyrate, that we've done for a number of different developmental and epileptic encephalopathies. I worry a little bit that I'm going to bore some people, but so I'll go through some of the logistics and some of what we did somewhat quickly, and then what I'm really interested to share with you is some of the developmental outcomes we've been looking at. For Marie and for other folks that think a lot about this, you're going to see me struggle, and that's a little bit intentional because I think we really need to work on this and figure out how to analyze this data effectively. These are my disclosures. This work is largely funded by y'all, that you guys started this. The way that we got started was through parent-raised funding through SLC6A1 Connect, through STXBP1 disorders, Clara inspired. The Orphan Disease Center was similar. And then once we showed that this drug may be effective, Horizon, which has now been acquired from Amgen, gave us some of the drug, and they've been very generous in supporting our work. This is our team. It's a collaboration between my group at Cornell and Scott Demarest and his team over at Colorado. It's been an incredible joy to work together with them. So what is this drug for phenylbutyrate? It's an FDA-approved drug for something else, for urea cycle disorders. It crosses the blood-brain barrier, and it comes in two flavors. The disgusting flavor is sodium phenylbutyrate. This is bitter. It's hard to tolerate. And then there's the, I guess, not so bad flavor of glycerol phenylbutyrate. I saw there are little bottles of it on your table, which is amazing. I thought that was spectacular.
Would you please add for $800 souvenirs?
They're $4,500. $4,500.
Everybody should arm wrestle over these. It could be a really long time. This is what it looks like. This is what gold looks like.
So, speaking informally, I was able to get a bottle for it because we were sort of thinking about a placebo. It doesn't taste so bad. If you've ever been in a pile of leaves, and it sort of tastes like being in a pile of leaves. That's how it. I don't know. That was my impression. At any rate, it's a liquid. It doesn't taste bad. We really have not had tolerability problems with it. So why this drug? Initially, Caddy Kang's lab and Jackie Bure's lab at my place. Caddy Kang is at Nashville, at Vanderbilt. And then several other labs had this idea that this drug might be effective for several different epilepsies. And so we were able to get a hold of it based on this bench neuroscience work. The collection of genes, I'll go through them. STXBP1 is involved in releasing synaptic vesicle contents into the synapse. SLC6A1 is a GABA reuptake channel. GAT1. GABA A receptor, they're around here. GABA A is sort of an inhibitory listener in neurons. LGI1, we haven't tried it in people with this, but there is some preclinical research suggesting it might be effective. It's involved in organizing the synapse between the pre- and postsynaptic membranes. SLC6A8, this is creatine transporter disorder. That gene may also be involved in synaptic activity. SYNGAP, they're also here, is involved in LTP and involved in synaptic plasticity. And SCN1A is the gene that causes Dravet disease, Dravet syndrome. What is the organizing principle? All of these are associated with epilepsy and seizures. They all impact synaptic function in some way or another. And all of them except for SLC6A8 are heterozygous, meaning you have one good copy and one bad copy. Millie Stone, there were two shout outs I wanted to give. We got a shout out for being the first study, maybe the first site to open for the natural history study. That is all Millie Stone and Natalie Wayland, our research group. The other shout out, and I'm sorry I'm interrupting myself, Jacob Tiller is amazing. Where is Jacob? So if anyone is on a medical school admissions committee, he's your guy. Okay, please. Just wanted to give you a shout out. I wrote him a recommendation, and I was like, listen, if he was a medical student, I would say he was the top 5 I've ever worked with. As an undergraduate, he is unsurpassed. It's really, anyway, Millie is also amazing, also applying for medical school if any of you are on committees. So Millie wrote a review for why do we think phenylbutyrate is working, and she listed multiple cellular mechanisms. Truth be told, we don't know which of these is right. For SLC6A1, Mike Cavanaugh has this idea that it's a positive allosteric modulator for GAT1. That kind of resonates for me because this drug works best for SLC6A1, which is a mutation in GAT1. But we don't really know, and people are looking into some of these other mechanisms. Regardless of what it does at the cellular level, functionally, you're haploinsufficient. So either it's making the wild type better, it's making the mutant work better, it's protecting the wild type from toxic effects of the mutant, or it's doing something else that we haven't thought of, and maybe just has a direct anti-seizure effect. We were convinced enough, we launched a trial. It was an open label, multiple dose, single treatment group trial. We screened people, we enrolled them, we got baseline for 4 to 6 weeks. We brought them in for 3 days and 2 nights, either at Cornell or in Colorado. Initially, we brought them back 6 weeks later, and then Dr. Demarest said, you should do a standard epilepsy trial, so we did 12 weeks in later parts of the trial. And then people had the choice to either come off of the drug or to stay on. Point of clarity, Amgen is extending our extended use period to everybody in the study. There's 50 people who we enrolled, and all 50 will have access through December 2026. Getting access to everyone else is high in mind, but those who are in the study, you can still have it through 2026 December. Safety and seizure results. We published a preprint. We are working on getting this to an actual print, but there is some data out there. And the safety and tolerability was really very favorable. No adverse events. The FDA was not surprised by anything that we found. Odor, there's a sort of a sweet odor that some kids get. They can get sleepy. They can have some appetite suppression. Metabolic acidosis has been a problem. There have been children hospitalized because of the metabolic acidosis. So it's not benign, but the FDA knew about this. So we didn't have to stop when that happened. We haven't really had a lot of missed doses. We've enrolled 50. 37 of those original 50 are still taking the drug. There were 10 that didn't work, and so they came off. There were 2 where it didn't work and they had side effects that were undesirable, so they came off. And then there was one child who came off because that child wanted to enroll in a different study. I want to think with you about development. We've heard a lot about Bailey and Vineland and all of these ways to think about development. The way I think about development for SLC6A1 and multiple other developmental epileptic encephalopathies is in this space. X-axis, chronologic age, y-axis, age equivalent. My 5-year-old speaks like a 2-year-old, right? Like we can kind of wrap our head around that conceptual space. A neurotypical child follows the diagonal. As they get older chronologically, their abilities age with them. You've got variations, but ultimately their chronological age and the age equivalent on these tests should roughly be the same. And so they'll track along the diagonal. A child with a DEE, remember these are developmental epileptic encephalopathies, developmental delay, epilepsy, and the seizures and abnormal EEG make the development worse. Clinically, they plateau. This is what we typically see. They make some gains, and then at some point, they stop. And you hear about small gains and little things that they do, but kids who by age 10 aren't walking, they're not going to start walking. If by age 10 they're not talking, they're not going to start talking. And so we have this plateau. In this era of new therapies, we want to beat the plateau. And so a therapy, it may not get you to perfect, but we want it to be better than you would have been. And so this blue trajectory is our goal. And so what you're now going to see is how do you take this and apply it to real data, which is messy. First, you have to do math. I was a math teacher before I went to medical school, and so it was kind of fun to be able to do this. So there's that dotted line neurotypical, and then like, how do we model this developmental plateau? We can use math. The fun thing about this equation, it's an exponential decay, and there's one parameter. There's this parameter k, and it appears twice. It's there at the beginning, and then it's underneath the exponent thing. What's nice about this is you only need one piece of data to find that k. That was intentional. It's very simplistic. This is probably wrong. But all models, I didn't know that was going to get a laugh. All models are wrong. So this one, we don't know how wrong it is. And I think one of the things that's going to happen for these natural history studies is we'll figure out what particular things about this are not correct. All right, so I'm doing the same talk for other disease groups, but this is you guys. So first of all, the seizure data. Anyone who's seen me talk has seen this slide. The results for SLC6A1 with Ravicti have been really strong. We gave it to 10 children. 7 of them we called a responder. And then 3 of them were like, they're not really not responders, but they had other things that prevented us from wanting to call them responders. And the effect has been sustained. There are children who have been on the drug since 2020 and are legitimately seizure-free ongoing. The clinical nugget that has sealed the deal for me is that I got frantic calls from families when the seizures came back, and we said, what's happening? The child had gotten bigger. And so as children grow, if it's a dose-dependent dose, you have to increase the dose. And what has happened consistently is when we readjust the dose for their weight, the seizures go away again. So that's my clinical interpretation, but it's been very compelling. So here's where it gets messy. There's not a lot of data out there about developmental trajectories for SLC6A1. It exists in some pockets that you've heard about, and I think one thing, Amber, that I am with you on is bringing this all together so we can learn from it. Simon's, Searchlight, and RareX have some data, so I pulled that out for Vineland in particular, and then fit that plateau, 3 minutes, fit that plateau to 4 domains: expressive, receptive, gross motor, fine motor. And you can see like, well, it's not too bad. And then those K values, they're around 2 years old. So most children with SLC6A1 in those 4 domains roughly are acting like 2-year-olds, even if they're 10, even if they're 15. Here's the data. This is where it gets messier. These are plotted trajectories for receptive, expressive, gross motor, and fine motor. What I did is rather than put the points, I made little line segments. The blue ones are ones where the developmental age equivalent is at least 3 months better than you'd expect based on the plateau. I made that up. I just want you to know that. But it gives you some sense of what we might be seeing. And so there's actually 5 little blue segments that are better than expected. And then we did it for the other domains. I think what's remarkable about SLC6A1 compared to the other disorders is that there's some super responders. Like, look at those. Like, some of those lines are steep. Some of those lines are headed back to normal. So that was really exciting to see. And the stronger we have natural history data, the more confident we'll be able to look at some of those trajectories and say whether this was just the disease does this, like some kids just sort of perk up, or whether this was the drug. And it's going to be the same question for whether it was the gene therapy. Limitations, small sample size, we only did 10, open label study. For SLC6A1, the blinking spells, the absences, the head drops are difficult to count. Next steps, we're going to write up the seizure outcomes. I've got all the diseases, so I'll write up the seizure outcomes for all 50 kids. The non-seizure outcomes will come next, hopefully supplemented by some natural history data so we can really make sense of the development. You heard Jacob give the amazing talk on EEG biomarker development. That got better in a lot of kids. I didn't show the slide today, but we had kids who came in with sort of EEG on fire, lots of slow spike and wave, lots of seizures, and then 6 weeks later, like it's gone with the Ravicti. And so we need to quantify that. It's not enough for me just to kind of say that and sound impressive. We have to actually measure it. And so Jacob's work is really about developing those quantitative techniques to track that change. And then we're working on compendia for insurance. So what insurance companies do when you say, I want this drug, is they look it up in a book, and if it's not there, then they may deny it. Many people have had some success getting Ravicti for their kids, but it's kind of hit or miss. And so we have submitted, there's 4 of these organizations. We've submitted requests. I thought, Amber, I feel guilty because it took us a while to do this, but I thought like you needed to submit this big packet. And so I kept asking people like, how do you submit a packet? And no one seemed to know. So I went to the websites and like, it's like a form. You're like, hi, can you add this indication? And then I got an email back like, thanks for your idea. We'll think about it. Like that's it. So I did that for all 4. So I'll keep nudging them, and then as these papers come out, we'll nudge them more. There are more papers coming out. Kristen Barber, who's my colleague at UCSD, spoke with many families, and she has some observational data on another 15 or so families. There's a group in Europe who's also working on a case series of about 5. So my hope is that as the literature builds and there's more arguments for this, it'll be easier to justify to insurance. And then Stefan is here. I've been thinking about Europe. I've been thinking about Israel. I've been thinking about sort of the rest of the world. Like, how are we going to get this data into people's hands and help form the argument to make this available to folks? Thank you.